You give the round of applause. All right, we have Professor Randy Schekman with us now. I will actually start from a group photo first. Can I invite all of you, Professor and the Dean, to stand up. So we're going to have a big group photo first. So the camera will be panning from the stage and then forward to you guys at the front, and the student at the back, can you stand up as well? So you can enjoy the moment in the photo. So yes, if you could squeeze in toward the middle line, that would be amazing. Yeah, that's perfect. Yeah, just squeeze in. We're getting there. Looks very good. Right, so 1, 2, 3, and cheese as well. 1, 2, 3. Okay, 1, 2, 3, cheese. Only cheese? Okay, please sit down. Thank you so much, everyone. And then will be maybe the video of this series. Yeah. Good afternoon, ladies and gentlemen. My name is Dr. Nawamin Bin Pathomrat. I am Assistant Dean for Education at the Faculty of Medicine, Prince of Songkla University. Today, I'm very pleased to welcome all of you to join this fifth ceremony of JAPAN-ASEAN BRIDGES event, facilitated by International Peace Foundations, and this event sponsored by Mitsui Fudosan Group, Dusit Thani Hotel and Resort, Kasikorn Bank, and also BMU Thailand. Today is a great honor for Faculty of Medicine and Prince of Songkla University to be the host of this special event. And this event will be presenting the talk from the Nobel Prize winner. The talk is on the topic of the role of basic science in biotechnology, provided by Professor Randy Schekman. He is a Nobel Laureate for Medicine in 2013. To commence our program, may I call upon Associate Professor Reungsak Reethanaporn. He is the Dean of Faculty of Medicine, Prince of Songkla University, to give his welcome remarks.
Esteemed President of University Council, Professor Jarat Suwanvela, esteemed President Dr. Niwat Kaopradap, Honorable Professor Dr. Randy Schekman, Nobel Laureate in Medicine, Mr. Uwe Morawetz, founding chairman of the International Peace Foundation, distinguished colleagues and ladies and gentlemen. With great pleasure and honor, we extend our heartfelt welcome to the fifth talk of ASEAN-Japan Bridges event series, co-hosted by the Faculty of Medicine, Prince of Songkla University. Your presence, especially that of Nobel Laureate Dr. Randy Schekman, illuminates a crucial role for medical science in fostering global peace and prosperity. At our Faculty of Medicine, our dedication to advancing healthcare innovation is rooted in a profound commitment to humanity's well-being. We are driven by the noble intention of utilizing our knowledge and expertise for the betterment of mankind, ensuring equal success to quality medical services, and fostering understanding and cooperation among diverse communities. Today, as we convene to explore the transformative power of medical science, we are reminded of the noble aspiration that defied the prestigious Nobel Prize. It's our sincere hope that Dr. Schekman's insight will not only inspire our medical students and faculty members, but also for all those who are present here, to intensify their efforts in advancing the frontiers of medical research and practice. We are deeply honored to co-host this event and extend our heartfelt gratitude for all who have contributed to its success. Without further delay, I would like to invite Dr. Niwat Kaopradap, President of Prince of Songkla University, to deliver the opening remarks. Thank you.
Esteemed Professor Dr. Jarat Suwanvela, Chairman of the University Council, Honorable Professor Dr. Randy Schekman, Nobel Laureate for Medicine, Associate Professor Ryungsak Lee Thanaporn, MD, Dean of the Faculty of Medicine, Mr. Uwe Morawetz, Founding Chairman of the International Peace Foundation, distinguished guests, ladies and gentlemen, on behalf of Prince of Songkla University, it's my great pleasure to join the fifth event under the JAPAN-ASEAN BRIDGES event series this year. Hosting this five auspicious event in collaboration with the International Peace Foundation has been an eye-opening experience, one that we will cherish and remember fondly for many years to come. The occasion also marks Prince of Songkla University's 55th anniversary this year, throughout which our university has been continuously leading in terms of innovation, research, and higher education, both in the southern region, nationwide, and international recognition. A large part of this outstanding success is due to our excellent Faculty of Medicine, which has been an integral part of Prince of Songkla University since its foundation. This is why I am confident that today's event, co-hosted by the Faculty of Medicine, will both increase our institution's global visibility, research networking, and reputation, and to serve as a significant milestone toward the betterment of society. I strongly believe that the insights we will gain from today's talk, entitled The Role of Basic Science in Biotechnology, delivered by the Honorable Professor Dr. Randy Schekman, will inspire our faculty, students, and all participants towards achieving greater goals and elevating our community to greater heights. Honorable Professors, ladies and gentlemen, on behalf of Prince of Songkla University, I would like to thank to Professor Dr. Randy Schekman, Nobel Laureate for Medicine, for sharing his inspirational wisdom with us today. I would also like to express our gratitude once again to PSU Faculty of Medicine for co-hosting this auspicious event, and of course, to the International Peace Foundation for trusting PSU to make the event happen. Finally, I wish to convey my sincere wishes that this talk would become a successful event and to inspire all of us, as well as to the youth of Southern Thailand. Thank you.
ครับ Thank you very much นะครับ Assistant Professor Dr. นิวัฒน์แก้วประดับนะครับ for his opening remarks. Next, I would like to invite Mr. Uwe Morawetz. He is the founding chairman of the International Peace Foundation to deliver his introductory remarks ครับ
Yeah, welcome to the Japan-ASEAN Bridges event series, which is facilitated by the Vienna-based International Peace Foundation. The events are hosted in cooperation with some of the main universities of this country, and I would like to thank Prince of Songkla University for hosting our and final event in this Bridges series today, commemorating the 50th anniversary of official relations between Japan and the ASEAN region. Bridges has been continuously held in Thailand and Japan since November last year, involving the participation of Nobel Laureates for Peace, Physics, Chemistry, Medicine, and Economics. The Japan-ASEAN Bridges series follows the series of 800 Bridges events, which the International Peace Foundation has facilitated since 2003 to support education in the ASEAN region. This peace cannot be achieved instantly, but is a process which needs time. Bridges has not been organized as one single conference, but as an ongoing series of events of now over 2 decades, in which Nobel laureates and international decision makers have built strong bridges with leaders in all parts of societies and with the general public. With the basis for peace being education and synergies being the fruit of cooperation, the International Peace Foundation has not realized bridges alone, but has carried out the program together with UNESCO and with 179 other national and international institutions, including 106 major universities and schools across the ASEAN region. The multidisciplinary and pluralistic approach of bridges in the Philippines, Malaysia, Cambodia, Vietnam, Singapore, Indonesia, Laos, and of the events now here in Thailand reflects that peace involves all parts of society. It involves awareness and social responsibility of politicians, its business community, scientists, artists, and the media. And since peace within ourselves, our families, and our environment starts in our minds and hearts, it involves every one of us. In this sense, Bridges challenges us to cross borders and to build bridges by listening and opening up to other viewpoints, by generating new thoughts, by developing innovative forms of cooperation, and by fulfilling the desire of everyone to get to know and learn from each other. This can lead us to a world in which we will be able to understand the complexities we face today in a new light. So let us be inspired by the knowledge and the wisdom that Bridges continues to offer. An opportunity to get a more inclusive, interconnected, and comprehensive view of ourselves and the world in which we live in, and which we are able to create anew constantly through dialogues towards a culture of peace which need the participation of all of us. I thank the 2013 Nobel Laureate for Medicine, Professor Randy Schekman, who has agreed to come to Thailand to support the events, and we now look forward to his speech and to his important contribution to build bridges. A warm welcome to Thailand and to the south of Thailand, Professor Schekman.
Well, good afternoon. Thank you for coming to join this occasion late in the afternoon. I'm delighted to be here. This is the third of my series of lectures in Thailand. I've thoroughly enjoyed all of my hosts, and I'm grateful to the Prince of Songkla University for sponsoring this event. I'm particularly grateful to my host for all this trip, and that's Uwe Morawetz, whose enormous efforts to bring this Bridges program to fruition over many years is benefited and enriched the educational environment here in Thailand and elsewhere in Southeast Asia enormously. Uwe, we are all grateful to you. So what I'm going to do today is to start off easy. I'm going to talk a little bit about what we know about how genes are expressed, what genes do. Many of you have already had basic coursework in this, but I think this is a rather broad audience. So I thought I'd start out slow and build to the latest revolution in genome editing, which has given us, just this past year, the first breakthrough in treating and curing a genetic disease. This breakthrough has only been possible because of a history of investment around the world in basic science, principally basic science focused on the study of how microorganisms function. For many years, this work focused on a single bacterial species, E. coli, but it then advanced to other bacterial species and to fungi and yeast. And now, with the tools that were developed as a result of this basic science on microorganisms, we have the possibility in our hands of treating genetic diseases that have afflicted humanity since the beginning of evolution. So I want to share with you that excitement of this discovery and application of this knowledge. So let's move forward. First of all, I'm sure you've all had in coursework the discovery of the nature of the genetic material. There was a rich history in finding first that the DNA molecule is the source of genetic information in the 1940s, but it was then in a major discovery in 1953 by Watson and Crick with assistance from other individuals, particularly Rosalind Franklin in London, that it became possible to understand that the genetic information is encoded by a double helical molecule, DNA, that consists of strings of nucleotides, adenine, guanine, cytosine, and thymine. and that by reading the sequence of nucleotides, one could eventually predict the sequence of amino acids that become encoded, that are encoded by a DNA molecule. It's also possible to understand, because of the way the bases, adenine forming 2 hydrogen bonds with thymine, and guanine forming 3 hydrogen bonds with cytosine, that the DNA molecule can be unraveled, copied, and duplicated. Thus, in a master stroke of discovery in 1953, we could appreciate how DNA could be perpetuated and passed on from generation to generation. That discovery and the excitement about the experiments that led to that discovery and followed from it launched an amazing period of discovery around the world in an area that was then and still called molecular biology. Now, once it became clear that the DNA was the source of information for the expression of genes, there were a number of experiments, very often conducted in Britain and in the United States, that led to the realization that DNA housed in a chromosome in a eukaryotic cell, a cell that has a nucleus, the DNA is housed in the nucleus, but in bacteria, the DNA molecule is free in the cytoplasm. It became clear that the DNA molecule is, in a sense, expressed, translated, by having an RNA nucleic acid copy made of the DNA. So an enzyme that's capable of unfolding the DNA inserts itself and uses one strand of this DNA duplex to make a copy in a transient form of a nucleic acid called RNA. And this RNA is then the same information as was in the DNA, and then that RNA somehow has to be recognized and the information transferred to the sequence of amino acids that result in polymerization of a protein. So this is indicated in really simple form here. There's a lot of detail that I won't bother you with, but we now understand from what was then called the central dogma that DNA encodes RNA copy, and the information in RNA can be translated to make a protein. Protein is a stretch of amino acids, sometimes thousands of amino acids long, and we know from basic protein biochemistry that the information for folding a protein is intrinsic to the sequence of amino acids, primarily the side chains of the 20 different amino acids, and the chemistry of their interaction dictates the final folded and functional form of a protein molecule. So that covers several decades of molecular biology in a very simple picture. Now, in the late 1950s, the great American chemist Linus Pauling, who had really imagined and done much of the experiment to elucidate the nature of the covalent bond, recognized in another really stroke of genius that genetic diseases may result in the change in the positioning of an amino acid in a protein molecule. If by changing one of the bases in the DNA through mutation, that information is somehow changed to alter the positioning of amino acid, you would end up with an amino acid that didn't belong there and might ultimately affect the folded structure and function of a protein molecule. I mean, it's almost a trivial idea that we can imagine right now, but it was revolutionary at the time. And then he set about in his laboratory at Caltech in Pasadena in California to experimentally demonstrate that one very serious genetic disease, sickle cell anemia, changes one amino acid in one of the subunits of the oxygen-carrying molecule hemoglobin. One of the subunits, a globin chain, has in sickle cell anemia a change from one amino acid to another that changes its physical properties. He was able to show this experimentally, and that was the first of what we now understand are a series, probably thousands of different genetic diseases that are the result of such mutations. Now, at the time, and of course, until now, it's been a dream that there might be some tool that would allow us to change such mutations back to normal. If one could simply go in to human cells in a living person and introduce a precise scalpel that would allow you to remove the wrong mutation and replace it with the correct nucleotide, you might be able to cure genetic diseases. Now, we're a long way from doing what I've just said, but I'm going to share with you in a moment that it is now possible, indeed in the past year, it has been possible to do that with sickle cell anemia. So the idea is, what if you could go into the text, as depicted here by some random language, if you could go into the text and read it and find the incorrect mutant substitution of an amino acid and then replace it, just like you can correct errors when you're typing a report. Well, now I'm going to tell you about the history of a discovery that has led to this revolution. There's an arms race going on in the world. It's been going on for 3 billion years. There is a race between bacteria and the viruses that infect bacteria. These are by far the most numerous living objects on Earth. It's estimated that in ocean water, every milliliter of ocean water has probably almost 1 billion different bacterial virus particles. So imagine that, multiply that by the whole Earth. And there's an arms race, bacteria trying to survive infection by a virus, and viruses evolving to combat these bacteria. In the course of this evolution, one particular method was stumbled on by chance, a primitive form of immunity that a bacterium has evolved to ward off and degrade foreign DNA. This primitive form is now what we now call the CRISPR-Cas9 revolution. Bacteria, and people studying bacteria over a period of several decades, were able to understand that bacteria encode a protein that can make a double-strand break in a foreign DNA molecule, and this enzyme that makes such a double-strand break is informed in making that double-strand break by a small piece of RNA, actually 2 pieces of RNA, that the enzyme uses to find foreign DNA that happens into the cell. So that's been discovered and worked on by microbiologists for many years. And these viruses, there are billions of different kinds of viruses. They are viruses that specifically affect certain species of bacteria. A virus such as you see in this diagram is one of a virus called T-phage viruses that infect E. coli. These viruses land on the surface of the bacterium, and they literally pump the viral DNA molecule into the cell, and the cell then is a host for replication of that viral DNA. And if the host doesn't have some way of warding off that virus, the virus will reproduce itself by the hundreds, and then the bacterium will explode and the viruses will spread around in a neighboring group of organisms. But as I said, the bacteria have evolved multiple different systems of immunity against bacterial viruses. So one form of immunity, as I mentioned a moment ago, is the use of RNA copied from the bacterial DNA and a specific sequence of RNA that has evolved to recognize and bind to an enzyme that will then cleave foreign DNA at a site that the bacterium recognizes not as its own. So RNA can have a function independent of protein synthesis in this example. And the discovery resulted in a revolution, part of which was achieved by my great Berkeley colleague Jennifer Doudna, who won the Nobel Prize 2 years ago with Emmanuel Charpentier of Europe, for their recognition that this enzyme called Cas9, which you see a structure of here, a space-filling structure, this enzyme Cas9 binds a particular so-called guide RNA, and the guide RNA finds its sequence on a foreign DNA molecule, and then the Cas9 molecule recognizes that sequence and chops the foreign DNA in half by cleaving both strands of a double helix foreign DNA, and that then destroys the virus. Now, it was recognized by Jennifer, by Emmanuel, and by many other investigators that this tool, this enzyme, might be used in a bioengineering approach to change DNA molecules, not just foreign DNA molecules, but change host DNA molecules in a deliberate manner. So, for instance, as you see in this diagram, the DNA may be cleaved in half by the enzyme Cas9, and one might then be able to use a piece of DNA that encodes something else to stitch in between where the break had been made. This might then be a replacement DNA, a piece of DNA that could replace a defective region that might be then the substrate that was cleaved in the first place. So that was the general idea, and this technique has now evolved over the past 10 years or so to be of direct practical application. Now I'm going to show you a little video that Jennifer Doudna shared with me that describes this approach in a little bit more detail.
Scientists have long imagined being able to make
Can you
specific
hear
changes
this?
to the genetic code of
Yeah.
a cell or organism. A revolutionary genome editing technology invented by scientists at UC Berkeley and Umeå University makes this possible using an RNA-guided protein called Cas9. Cas9 searches through the cell's vast genetic material until it finds a sequence of DNA that matches the sequence of its programmable guide RNA. The guide RNA enables Cas9 to open the DNA helix and to position two molecular blades to cut each strand of the DNA double helix. Repair enzymes seal a gap in the broken DNA, sometimes by inserting new genetic information supplied by the cell or the experimenter. In this way, disease-causing mutations can be corrected by changing the underlying genetic code. Scientists are now using Cas9 technology for research applications in human health, agriculture, and bioenergy.
When Jennifer Doudna published her revolutionary paper in 2012, that unleashed a storm of interest in using this technology for deliberate genome editing opportunities. And so what kinds of genomes could we access with this technology now 10 years ago? Well, initially, it became possible to use this as an experimental tool to manipulate cells or small mammals like mice, or perhaps even non-human primates. But eventually, the goal would be to use it to engineer mutations in human genetic diseases. But of course, the controversy then emerged almost immediately about which kinds of genetic traits would we wish to change. Obviously, changing the mutant DNA in genetic diseases would be particularly important. But if we had the prospect of engineering other aspects of human personality or behavior, that brings some quite serious moral and ethical issues. So let me tell you about two examples of the extremes of the application of this technique. The first is that about 6 years ago, when it first really became feasible to try to alter the genome in a human embryo, most scientists around the world thought this would be a terrible idea, that one should avoid messing around with the germline and changing an embryo. But a physician in China realized that it might be possible to create a human embryo that was intrinsically resistant to infection by the AIDS virus. We knew from decades of study of the AIDS virus that there's a particular receptor on the surface of immune cells to which the virus particle adheres as it enters the cell and expresses its own nucleic acid. And there are some humans walking the world who have a natural resistance to HIV because they have mutations in the gene that encodes that receptor molecule. So this physician thought it would be a great idea to use this CRISPR-Cas9 technology to change that gene in the embryo of two embryos that were going to be implanted into a mother who wanted to have children. And he did this without any supervision other than in his own laboratory. And two children are now walking around, they're probably still young, who have had that gene, the receptor for the AIDS virus, altered so that they could never become infected by the AIDS virus. Now that sounds great, doesn't it? But the problem is that the technique is not 100% precise. And so it's quite likely that these two children have unexpected mutations that happened elsewhere in the genome. And so as a result of publicity about this discovery, there was a firestorm of criticism that resulted in this Chinese scientist losing his laboratory and now not being able to do clinical science ever again. So there is now a moratorium on the use of this technology, at least in treating human embryos, to try to change personality or even any genetic defects. Now, of course, there's another way of doing this, and that is possibly by taking white blood cells out of a patient and evicting the HIV receptor gene in white blood cells and growing those cells up in the laboratory and then infusing them back into a patient. And that probably is safer because you aren't affecting the embryo, you're affecting adult cells. That particular kind of application has now been achieved just in the past year by a scientist at Harvard Medical School and a company in Boston that has found a way to cure sickle cell anemia. So let me tell you a little bit about what they did. In the human, babies, or before they're born, produce a form of globin that is different than the adult form of globin. It's called fetal globin, and that fetal globin produces a hemoglobin molecule that has a higher affinity for oxygen so that the fetus can absorb oxygen more readily from the mother. When the baby is born, that fetal globin gene gets turned off by another gene that encodes a kind of a repressor that keeps the fetal globin gene from being expressed in an adult. At that same time, at the same region of the genome, adult globin begins to be made. And so we all in this room are expressing and relying entirely on adult globin. So this scientist at Harvard Medical School, imagine, what if it were possible to evict, to mutate the gene that normally turns off the expression of fetal hemoglobin in an adult, particularly in adults suffering from sickle cell anemia? What if it were possible to do that? Could we then allow fetal hemoglobin to be expressed in a sickle cell patient, replacing the mutant adult globin? Well, as it turns out, it works. It's now been applied to about a dozen patients who are now basically restored to health because they are relying now, unusually, but relying on fetal globin expressed now in their adult white blood cells, whereas the now mutant globin is no longer being produced. So that's the first real breakthrough of this technology and shows tremendous promise. That kind of very targeted approach, I think, is going to be the future of this kind of technology. Now, as I said, we still continue to use this technique, and we can manipulate the germline in experimental animals. So as a tool for science, it's become really essential. Everyone who studies mammalian development in experimental animals relies on this kind of germline editing to study the role of genes in organisms as they develop. And it continues to be very controversial as in its application. There are many other kinds of applications that I'd like to tell you about now briefly that many of us feel are worthwhile, aside from the moratorium on editing the genome of an embryo. There are other applications in biotechnology that are particularly valuable. aside from engineering a baby, there are other methods and other things that really merit the application of this technique. And one is in the enormous need to improve sources of agricultural products around the world. The population of the world continues to expand. Regions of agricultural productivity are subject to climate change, the rigors of climate change. What if we could use these tools that I've described to make genes that are more resistant, that produce plants and agricultural products that can grow in climates that have less water availability or higher temperatures? Or could we produce plants that have additional nutrient value that would sustain populations that are otherwise reliant on a limiting agricultural product? So one such example that has already reached the market is an improved mushroom that's now been produced by using CRISPR techniques to produce mushrooms and other plants that are resistant to fungal attack. These products are now on the market, and certainly in the United States they're very promising, and they've generally been accepted. But in Europe, there's been a kind of revolt against the use of these recombinant techniques in agriculture because farmers want traditional techniques for genetic breeding, which are much less precise than the tools of CRISPR-Cas9. And so there's been a struggle between those forces of agriculture that want to use biotechnology and those that oppose it. One particular example that is of interest and concern here in Southeast Asia is the use of a technology that had been developed for many years to engineer a form of rice that allows the rice to also produce vitamin A. Vitamin A deficiency in the world is of greater concern than even AIDS or tuberculosis or malaria. In Southeast Asia, among subsistence farmers, there is a paucity of supplements for vitamin A. I mean, people in the Western world can take vitamin supplements, but around the world, people who are relying exclusively on their own agricultural products do not have adequate sources of vitamin A. And so, engineers, biomedical engineers, agricultural engineers produce a form of rice called golden rice, into which genes from other organisms, microorganisms, have been introduced to allow rice to produce vitamin A for nutritional supplementation. This rice has been tested in agricultural fields. It's been shown to be safe over many years, but it has generated a great deal of opposition from those organizations that feel that agricultural products should not be subject to biotechnological manipulation. So Greenpeace is one such organization, an organization that generally has been very supportive of the environment, but has taken, I think, an unrealistic position in opposition to the use of biotechnology in any agricultural application. In spite of this, there has been an enormous outpouring of support among health agencies, agricultural agencies, scientific agencies, and most scientists, certainly most life scientists, recognize the value of using biotechnology to enhance agricultural productivity. The opposition has come from very limited but very vocal groups, such as Greenpeace. Whoops, what happened here? Okay. Earlier this season, you had a lecture from Rich Roberts, who led an effort to line up 100 Nobel laureates, including myself, to sign a petition in support of the application and field testing of golden rice in Southeast Asia and around the world. This was opposed by Greenpeace, but this hit the news last year, and there was a news press conference, and many of us were very supportive and talked to our local media. Greenpeace went on the attack, and I was personally subject to harassment by an organization representing Greenpeace, who requested all of the emails that I had shared with Rich Roberts and others in preparation for signing this document. Fortunately, they weren't able to get that information, and the petition still stands as a strong testimony in favor of this and other biotechnological applications. Now, I'd like to turn for the rest of my talk to a very different kind of biotechnology, one that has arisen in the pharmaceutical industry, in the use of microorganisms and cells grown in culture to produce the new biological molecules that are increasingly important in therapeutic applications. One of them happens to be an application that came from some of my basic science conducted in my laboratory at the University of California, Berkeley. So before I tell you about that discovery, I want to give you a little tutorial on biological membranes and how biological membranes are involved in producing molecules like insulin in the pancreas that are secreted from pancreatic beta cells. So this is a cartoon of a piece of a biological membrane. Membranes surround all of our cells, and in almost all of our cells, except red blood cells, membranes surround intracellular compartments, compartments like the mitochondrion, or the nucleus, or the lysosome. And they all consist of a bilayer, 2 layers of these red molecules called phospholipids. Phospholipids have polar head groups, polar hydrophilic head groups that face either the outside of the cell or the cytoplasm, and they are covalently attached to long chain hydrocarbons called fatty acids, which are very apolar, hydrophobic. So when phospholipids are simply mixed together in a solution, they spontaneously form bilayers so as to satisfy both the apolar surfaces facing the aqueous environment in a buffer and the polar surfaces facing the aqueous environment and the apolar surfaces facing each other hidden within the bilayer. A bilayer like this by itself surrounding a cell wouldn't work because cells have to selectively take up charged ions and small molecules. So cells have evolved over 3 billion years, specialized proteins that often span the bilayer, shown here in green. These proteins may be channels or permeases through which molecules can pass in one direction or another, or they can be molecules like receptors, such as the insulin receptor. Insulin binds to such a receptor, and it causes a muscle cell or a fat cell to swallow up glucose that's just been produced during your digestive process. So all these membranes have this fundamental unit as part of their structure. Now, of course, cells differ quite dramatically in the membranes that they use to allow cells to function. Here is one such cell, a very complicated cell, one that's very important to all of us. It's the beta cell of the pancreas. It's the seat of synthesis and secretion of insulin, the hormone insulin. Insulin is housed in these many granules that you see in this thin slice through a beta cell. The insulin forms a crystalline complex in the interior of a membrane that surrounds the insulin. These are called insulin-containing granules. In between meals, as you're no longer digesting anything, insulin continues to be made. It gets stored in these granules, and it waits until you have another meal. And when you start digesting polysaccharides, this insulin is exported outside of the cell. Now, I've talked to you at the outset about the human genome. The human genome codes for about 23,000 different protein molecules. We know from all the data that all of these proteins are made inside of cells. They're made by little machines called ribosomes that stitch amino acids one next to another. All these proteins get made inside the cell, but some good fraction of them, maybe a quarter of them, have to escape. They have to be exported outside the cell by a process called secretion. And that process requires that molecules like insulin and many of the other molecules that get exported must somehow be recognized as different. They must be distinguished in some way from those proteins that are going to remain inside the cell. And part of that involves their selective encapsulation within a membrane to create a granule such as this. This process, on close inspection, results finally in the secretion of insulin dependent on a particular event at the very end of this process that is promoted by merging of membranes at the cell surface. Here is a close-up of the image I just showed you. This is an insulin granule in a cell that's about to be discharged from the beta cell. The membrane that surrounds the insulin granule reaches and touches the inner cytoplasmic surface of the plasma membrane, and at a key moment that's tightly regulated, these two membranes merge by a process called membrane fusion, and the insulin granule is ejected. The insulin crystalline form dissolves and it courses through your body and is recognized by receptors on muscle and fat cells that cause these cells to swallow up the glucose and store it for later use. This step of membrane fusion is a very important central event in virtually all the cells of your body. None more important than at the junction between a nerve cell and a muscle cell, or in the brain, the connection between nerve cells that transmit the information that is responsible for every aspect of our personality, of our thought process, of our memory, and of our sensations. This communication between nerve and muscle cells and between nerve cells is fundamental to our existence as a species. And it is dependent on this key membrane fusion event. Here at the junction between a nerve cell and a muscle cell, the nerve terminal has a collection of smaller granules. These are called synaptic vesicles. Synaptic vesicles don't have insulin. They don't really even have enzymes. They have instead a high chemical concentration of neurotransmitters, dopamine, serotonin, acetylcholine. Some of these vesicles migrate to the perimeter of the cell and dock. They sit on the surface, bound through protein interactions. And at a key moment, when these cells are stimulated by changing the membrane potential on the surface membrane, these vesicles are caused to fuse by membrane fusion, which causes the chemical neurotransmitter inside the vesicle to be discharged into the space, the synapse, between the nerve cell and the muscle cell. The chemical neurotransmitters diffuse across this gap, bind to receptors, in this case on the muscle cell, and that causes the muscle cell to contract. This is going on all the while, millions and millions of cells in your body. In the brain, there are tens of billions of nerve cells. Each nerve cell can make contact with 100 or up to 1 million different nerve cells, and each of these is involved in a very complex pattern of communication. That communication is indispensably dependent upon controlled fusion of synaptic vesicles at the synapse. This has been the subject of great inquiry by neurophysiologists and cell biologists over many decades. Why is this not behaving? Well, about 50 years ago, a great scientist in New York, George Pilati, was able to chart the path that all animal cells use to produce molecules like insulin and to move them from station to station within a cell that has a nucleus. And I've summarized Pilati's discovery shown here. He was able to show that ribosomes, those machines that stitch amino acids next to one another, those ribosomes in a pancreatic cell that's busy making lots of enzymes for secretion, those ribosomes are bound on the cytoplasmic surface of a canal-like network within the cell called the endoplasmic reticulum. The proteins that are made thread across a membrane, across the ER membrane, and they fold to make secreted proteins in this salmon-colored interior space called the lumen. This lumen then collects those molecules, such as insulin, and they diffuse into regions where they are pinched off into vesicles, which then are processed by further traffic through a structure called the Golgi apparatus, which was discovered by a 19th century Italian neuroscientist. Pallotti was able to show that this is an essential step in the movement of enzymes and proteins, like insulin. They are then further packaged into granules and delivered to the cell surface, where by membrane fusion they are exported. Now, I was introduced to this work when I was a graduate student, and I learned about Pallotti's work more directly when I was a postdoctoral fellow. But when I began my career in 1976 at the University of California, Berkeley, I wanted to study the mechanism that operates this pathway. When Pallotti won the Nobel Prize for discoveries in 1974, not a single protein molecule or lipid had been shown to play any particular role in this pathway. The mechanism of this process was completely unknown. We didn't know about a channel in the ER membrane through which new proteins would pass to enter the lumen. We didn't know how these vesicles could collect cargo molecules and pinch or move through the Golgi apparatus, or how they could migrate to the cell surface and fuse. Nothing was known about protein or lipid catalysts of this process. And so I began with a different approach. At the time, cell biologists studying this essential physiologic process relied on mammals, typically experimental animals, or cells grown in culture from human tissue or from animal tissue. And at the time, the tools to discover mechanism, the biochemical tools and genetic tools, were simply not available to study mammalian cells grown in culture. So I decided to use a different approach, using a simple microorganism, baker's yeast, Saccharomyces cerevisiae. Baker's yeast was at that time already a well-established experimental organism for the evaluation of processes that are essential to the growth of eukaryotic cells, that is, cells that have a nucleus. Yeast has a fundamental connection to our cells. We also have cells that have a nucleus, and yeast does too. And I hope to convince you in the next few minutes that the process that we use to make and secrete insulin is fundamentally the same as yeast cells use to make their own proteins. Yeast cells can be grown in the laboratory very conveniently. They have a generation time of about 90 minutes, so if you put them in a liquid broth that is full of nutrients, they will grow and divide logarithmically over many hours. During the course of the 90 minutes or so that it takes a yeast cell to grow, the way it grows is it sends out a bud. This small bud gets bigger and bigger during that 90 minutes, indicating that new surface material is being added to the bud. After about 90 minutes, the bud approaches the size of the mother cell, at which point a division plane is introduced and the two cells eventually separate. And if the nutritional conditions are good, this process can go on forever. Now, the virtue of yeast is not only that it's convenient, that you produce genetically identical cells, but it has a very convenient way of manipulating genes by traditional genetic analysis. Yeast cells can grow, unlike our cells, yeast cells can grow with a single copy of all of the 6,000 genes. It's said, therefore, to be haploid. It can also grow as a diploid, as our cells do. Our cells have 2 copies of each gene. But if you grow yeast cells in the haploid lifestyle, and you expose those cells to a chemical that randomly causes mutations, you will produce mutant genes that then will express some characteristic that you can evaluate by growing cells on a Petri plate. Now, of the 6,000 or so different genes in the yeast genome, about 1,500 of these genes are essential. So bear that in mind as I describe to you what it looks like inside of a yeast cell. So here's a thin slice looking through a yeast cell section using the electron microscope. Looks very different than a pancreatic beta cell, but the machinery is all there. There's a dramatic abundance of these ribosomes that are the machines that make protein molecules, but they're also membranes. This is an organelle that is similar to the lysosome in our cells. This is a strand of the endoplasmic reticulum in this particular section. Other sections you can see a nucleus, just like in our cells. But what the attention of my laboratory initially was drawn to this cluster of small little membrane vesicles, very similar in a way to the synaptic vesicles that I showed you a moment ago. Of course, these vesicles aren't secreting insulin. They aren't secreting chemical neurotransmitters. They're producing and secreting the proteins that a yeast cell needs to grow in the environment, such as in the surface of a grape. But one of the first predictions that we made was that the membrane of these vesicles could become the building block for the growth of the plasma membrane of the cell. So as these vesicles merge with the plasma membrane, step by step, the bud would get bigger. And thus the production or movement or fusion of these vesicles would be an essential process to allow the yeast cell to grow. That is, if you had genes required for the process of making these vesicles, and you interfered with the function of these genes by having a mutation, those cells would die. They wouldn't be able to grow because the vesicles couldn't make the bud get bigger. Now, microbiologists have a way of dealing with such genes and discovering their function, because if you just kill a gene, you produce a dead cell, and then you don't have nothing to work with. So what microbiologists have devised is a way of finding mutations, random mutations, that cripple a gene selectively. And one convenient way of selectively mutating these genes is to produce a form of the gene that works at, let's say, room temperature, but doesn't work at human body temperature, 37 degrees. And the way these mutations work is sometimes by random chance, if you expose a gene to a chemical that causes mutations, you'll change an amino acid that will render the protein product thermally unstable in a way that would allow the protein to work at a lower temperature. But if the temperature of the yeast culture is warmed up to 37 degrees, that protein will unfold and lose its function. Those are mutations that are called temperature-sensitive lethal mutations, and they've been used by molecular biologists for many years to discover essential processes of chromosome replication, of gene expression, of cell division. And this was our goal then, to use such an approach to find the genes that are required for protein secretion in yeast. So here's the graduate student in my lab who was responsible for the discovery that I'll tell you about. His name is Peter Novick, and you'll note, as was characteristic of those years, he had long hair. I had long hair back then, but I'm afraid it's mostly gone now. But nonetheless, he was a brilliant scientist, and I was very fortunate to have him in my lab. He is now a very successful scientist in his own right, and by chance, he is now the George Palade Chair of Cell Biology at the University of California, San Diego. Now, in late 1978, Peter isolated a temperature-sensitive colony of yeast cells and was able to show that when these cells are warmed up to 37 degrees, the enzymes that would normally be secreted from the cell build up inside the cell. And then when the cells are cooled back to room temperature, those enzymes built up in the cell are then secreted. So there's a thermally reversible effect of a mutation on a gene that we called SEC1, short for secretion. Just after we isolated the mutant, just by chance, the pioneer of this field, George Palade, visited Berkeley for 2 honorific lectures, and I was pleased to tell him about our work, but more importantly, Peter and other graduate students had dinner with Dr. Palade that evening. Peter shared the preliminary results with his mutant, and Palade said, well, now, of course, you must inspect these cells by electron microscopy. So we raced ahead to do this, and one of the great moments in my career came when Peter called me in my office at the top of our building down to the basement to look in the electron microscope at the following image. This is the cell, or one of the cells, that has this mutation, SEC1. Remember the image I showed you a moment ago. Cells have very few vesicles in the bud, but when this cell is warmed to 37 degrees for a couple of hours, just as it's about to start dying, these cells fill up with thousands of these vesicles. No longer restricted to the bud, they spill out and fill the entire cytoplasmic compartment, and that's because the SEC1 mutation cripples a gene that's required for these vesicles to fuse at the plasma membrane. So the vesicles continue to be made, they fill up the cell can't get any bigger, and eventually chokes itself to death. Now, over the course of the next few years and following many subsequent years of investigation, we isolated many more such genes, and I'll just summarize that in this picture. We were able to reveal a pathway in yeast, in baker's yeast, that is fundamentally the same as what Palade was able to show in pancreatic cells in mammals. But in this case, unlike what was possible in the pancreas, we could identify each step in this pathway with genes and therefore protein molecules. These protein molecules must catalyze some aspect of membrane fusion or protein traffic within the pathway. Now, it then became possible to clone these genes, to sequence their DNA, and to predict the amino acid sequence of the protein products. We did this for many of them, but the genome database at the time didn't have any genes of similar sequence because these were new genes that hadn't been discovered in the laboratory. But within a very short period of time, the human genome was sequenced, and all of the genes that we discovered are found in our genomes. In fact, SEC1, that is responsible for the end of the pathway in yeast, is found in all of our nerve cells, indeed all of the cells in our body that have a nucleus. But in nerve cells, that SEC1 protein is operating to engage synaptic vesicles to fuse at the presynaptic membrane to execute the process of neurotransmitter secretion. And that illustrates one of the last points I'll make, and that is, if I'm able, there is a commonality in biology that is uniformly informed by the process of evolution. I use Charles Darwin, a picture of Charles Darwin, to illustrate that the cells in Charles Darwin's body are fundamentally the same, with the same mechanics that yeast cells use to operate processes such as secretion, cell division, chromosome replication. As a result of this functional conservation, the biotechnology industry, growing up in the San Francisco area, was able to use yeast as a platform for the manufacture of large quantities of clinically important proteins. And so in my last point, I show you an example. One of the companies in the Bay Area, I was able to help introduce the gene for human insulin into yeast, and the process by which insulin is secreted in the pancreas being fundamentally the same as the process of protein secretion in yeast, it was possible to engineer yeast cells that produce vast quantities and secrete vast quantities of human insulin. Grown in large fermentation plants around the world, 1/3 of the world's supply of human recombinant insulin is made in genetically engineered yeast. Applications such as this are the result of intense basic science that is the foundation of industry and healthcare, and it is the origin of the biotechnology industry. All the work that I've described during the past hour is a result of basic science where people driven purely by curiosity make fundamental discoveries that will be applied in industry. So I urge those of you who are interested in science to first focus on basic science, because that will be the source of all new discoveries of clinical application in the future. Thank you for your attention.
Wow. Thank you so much นะครับ Professor Randy Schekman. I think this is very inspiring. I'm pretty sure that all the guests in this room is inspired and motivated by your talk as well. นะครับ So now we're going to move to the Q&A station. So please remain on the stage.
Of course.
And then please welcome all the questions from you guys. If you have any question related to the academic, or it can be anything related to the Nobel as well, please provide the questions. Can be in Thai and also in English as well. ครับ Any questions?
It always takes one brave soul to ask the first question. Others will follow.
Yeah. So we've got one question here as well.
There's a microphone in the aisle.
Yep. So
Oh,
please.
wait, somebody's coming to you with a microphone?
Yeah. Perfect.
Hi, thank you so much, Dr. Schekman, for the excellent talk from the Department of Epidemiology. But my question pertains to the CRISPR and the ethical implication. And I'm thinking more about, so I don't know if you've seen the movie Gattaca,
Yeah.
but okay, so I'll skip to that. I think the process in the movie about how kind of in the future world they breed this new group of humans is based not on CRISPR genetic modification, but it's similar to, I think, genetic medicine.
Yeah.
Like, they look at, okay, what are the best sperm and eggs parents can have, and then they breed a new set of humans who are in different social class.
Yeah.
Do you foresee this happening in the future, and to what extent? And what do you think are the ethical implications of the Gattaca world, for lack of a better term?
Well, technology always has unintended consequences. It's up to us as basic scientists to discover the natural world. It's up to society to decide how our discoveries are used. I've illustrated the problems that already emerged with the CRISPR technology, the inappropriate use of this as yet imperfect technology to engineer a human embryo. As a result of that one episode, there was an enormous firestorm of concern and criticism around the world. So now there is a very effective moratorium on the use of this technology until it's further perfected in the human embryo. And I think, Gattaca and any other thing that you might imagine, there must be input from society in deciding how this technology is used, which means that people in society must be educated to know what the technology is. There needs to be a greater appreciation of what science is and what science can do and what its limitations are. So I can't predict the future, but I know that an engaged and informed society is essential to control the misapplication of basic discovery.
Thank you.
Well said. Wow. Any questions coming? Well, I got one question personally. Is that okay? As a vaccinologist, and think this is very inspiring for you to use the gene editing yeast to produce insulin. Is anyone out there using gene editing yeast as a vaccine platform?
Gene editing of yeast as a
vaccine.
A vaccine, yes. Okay,
Vaccinology.
so in fact, the company that I helped engineer the secretion of human insulin also at exactly the same time found that if they took the gene for hepatitis surface antigen, which is the source of what has been in the past was a vaccine, if they took that gene and introduced it into yeast, that gene encodes a membrane protein, so that protein was inserted into a membrane, and it then accumulated in little vesicles without infectious nucleic acid. And now the entire world supply of vaccine against hepatitis B is made from vesicles that are isolated from broken yeast cells. So how many of you have had hepatitis B vaccine? Many of you. It's in fact a really very serious endemic problem in China. You've had little vesicles that were manufactured in yeast, and the fact that you've been vaccinated has just by itself been has reduced your likelihood of developing liver cancer. 90% of the cases of liver cancer in the world are as a result of a persistent Hep B infection. So this has been a terribly important vaccine. It's
Wow.
the one vaccine in the whole world for Hep
It's
B.
one of your contribution to the world again.
Well, I didn't do that, but I helped the company do this.
Amazing, amazing. Any questions? Yeah, perfect. Is any microphone?
He's got a microphone.
Yeah, perfect.
Good evening, Professor Randy. I am Patipanta Prik from Faculty of Veterinary Medicine. I am veterinary student here, only one here. I would like to ask you about the aspect that you have in the veterinarian treatment. As you can see, nowadays we have the high prevalence of rabies and in animals. Can we treat it with CRISPR-Cas9 because this virus will integrate into the naked body in axon? So.
Yeah. Well, I can't tell you right off the top of my head about using that particular technology, but one other technology that emerged in the pandemic is the use of mRNA vaccines. So it seems quite likely that one could have an mRNA vaccine, which is a piece of RNA, let's say, that from rabies virus coat protein that could be expressed and used as a rapid source of immunogen. But unfortunately, when animals or humans get infected, there has to be an immediate treatment with antibody. You can't wait around for an immune response. But one might be able to be vaccinated against rabies. And in the veterinary clinic, you could maybe treat animals to produce the vaccine so that they would be resistant to rabies infection. It's actually a direct application of the mRNA vaccine technology that we've used for coronavirus.
Unfortunately, some of them, some of cases in veterinary teaching hospital of Songkla University, they don't know that their pets, even they keeping pet inside home, they still get the rabies from their pet, and this is very sad because if they don't show sign, so can we treat it?
Well, I mean, to treat a rabies infection, you need an immediate source of an antibody. You can't wait around for an to elicit immune reaction.
If it's too late.
Yeah, well, so no, I mean, the way rabies is treated, if a human is bitten by an animal that is found to have rabies, they immediately get an infusion of anti-rabies antibodies. And that, for human treatment, is effective. But I'm suggesting that if people who are constantly being exposed to animals that have that infection, they could be immunized by an mRNA vaccine that encodes the rabies surface protein and thus have perhaps some durable resistance to infection.
Let me ask the second question, please. I'm curious about the bacterial phage that you have mentioned before, because I'm kind of very interested in bacterial phage. That is this could be used to apply with the do you know AMR
as a source of treatment of bacterial infections,
resistant the bacteria which is resistant to the antimicrobial. Can you treat it?
So antibiotics are losing their effectiveness because bacteria evolved their resistance mechanisms. But there's a new approach that is proving successful in the clinic, and that is to cultivate thousands of different species of bacteriophage and have a library of bacteriophage in a clinical laboratory. And then when someone has a persistent bacterial infection that is resistant to antibiotics, that bacterium can be cultivated. And then one can explore a library of different bacterial viruses to find one that works on that particular pathogen, propagate that bacterial virus in the laboratory, and then use it to infuse into a patient to kill the bacteria. This is now an emerging new approach that is sort of orthogonal to using antibiotics. And I think it has great promise because there are millions and millions of different bacterial virus species that can be cultivated in the laboratory. And if you have a big enough library of them, you're likely to find one or more that will be useful to treating a pathogen that is no longer sensitive to any antibiotics. In fact, there are clinics in the US that I know that are doing this.
Yeah, perfect.
So-
We still have the question lining up here as well.
Okay.
So if I
Thank you.
may, well, we have a lot of questions in the room. I'm very
Okay,
glad.
good. Well, it always
One
takes that first one.
one from the medical student here, and the second one will be from you,นะครับ. Yep. The mic is coming. It's traveling. Right. Okay.
Okay, good afternoon, sir. I'm still fifth year medical student. So I have a question about, in your opinion, how many years this technology like you use it to treat like the genetic disease such as the sickle cell or something like that? How many years it can use to apply to other country in the world, like
Yeah.
how many years it
Yeah.
can be used generally like that?
Yeah. So the example that I showed that I mentioned that just is from this past year in treating sickle cell anemia is a very dramatic example of a very specific disease that now can be cured. But it's enormously expensive to do this. And it is inaccessible to most places around the world. Most sickle cell patients are in Africa. Africa simply doesn't have the money. It's estimated, the physician who devised this technology told me he estimates that the procedure costs about $2 million. Now, of course, for a patient to live a whole life of sickle cell anemia is also very expensive, but that is not accessible to most places in the world. But the technology is going to advance. It's going to be much easier than what is now needed to cure a disease like this. For example, it will be possible to produce an mRNA vaccine that has an RNA molecule that encodes an antibody, for instance, that will activate a gene, or encodes a form of Cas9 and a guide RNA that can be used to introduce a specific mutation with great precision into the genome, instead of having to do the heroic genetic engineering that was necessary for treating this disease. So stay tuned. I am confident, and I know that Jennifer Doudna has a whole institute at Berkeley that's refining this technology. I'm confident that versions of the mRNA vaccine will be used in the not too distant future to treat genetic diseases without having to, in this case, take blood cells out of a patient. It'll be possible to just introduce the mRNA lipid nanoparticle directly into a patient. So I'm confident that this is going to develop in just a few years ahead.
Thank you.
Thank you. Any more questions from the area? Okay, we have this gentleman waiting to ask the question as well. Could you please standing up a little bit? Yeah.
Of course.
Perfect.ครับ.
Hello, Professor. I am a second year medical student. I just wonder, as you had said before about GMO technologies in the crops, that you have used Cas9 CRISPR technologies and it cuts the DNA in the crops. I was wonder is it possible that there is a small chance that the Cas9 protein will remain in the human bodies and it may cut some human's DNA in the future and may cause some mutation?
You mean the Cas9 having been used to create a plant? Would it be consumed by a human? And that seems highly unlikely. Highly unlikely. But plant breeding is basically a genetic technology that has been around for 100 years, is much less accurate because you're exchanging whole genomes between plants than this precision scalpel that's introducing a change in a particular place on a genome. So the technology is, to me, inherently much less problematic, less dangerous, less fraught with unexpected, unintended consequences than traditional plant breeding techniques.
So, in your opinion, they might have less likely to cause cancer or something in the future?
Well, I mean, you know, you can't say with any certainty that nothing untoward could happen. But it seems to me that the approach is highly unlikely. And you know, when products of genetic engineering in agriculture are produced, they have to be tested, and they have to be tested extensively. And these tests will reveal if there are problems. But this kind of technology is much more precise than what's being done now to breed plants.
Thank you very much, Professor.
Sure.
Thank you. Right. We still have time for a couple more questions. Wow, we have a question at the back there. Okay. Okay, so this question first, and then the second question will be yours, nakap. Yeah, go ahead. Yeah, please standing up and ask the question, please.
Okay, thank you very much for this. Good evening, everyone, and also with Professor Randy Schekman. While you're the first year medical students of Prince of Songkla University, it is such a great opportunity here for attending for this session, for all your sessions, as is not quite often here for everyone to visit and watch and have your lectures right here. I have two questions that I still wonder here. I'm wondering that one of the topics that you said and you discussed is about the ethical and moral issues of editing of some genes or genomes, such as anti-HIV resistance into someone's genes and might affect of the body's immunities or response. I would like to ask you that, is this possible, or is there any possibilities of one day all of these technologies and of performances might increase the chance of precision or increase the chance of success and decreasing of moral issues?
Yeah. Well, as I said, there is now a moratorium on doing this in the human embryo because of the concern that the technique is not 100% precise. But there's a great deal of work to try to improve the accuracy with which the Cas9 is delivered to a particular nucleotide base pair. And so as time goes on, it's going to be, nothing will be perfect, but it'll be much more accurate. So I'm confident that eventually it'll be possible to treat genetic diseases, although I think it'll still be a long time before we want to do it on the human embryo. I think it will be much safer to do it on somatic tissues that are at the root of particular genetic diseases, rather than exposing an embryo to changes that might affect the whole body during development.
Okay, that's a good one. I have one last question of mine, and might be the most beneficial to everyone as well, especially for med students. What is the principle of achieving Nobel Prize? Because in Thailand, there is no one achieving this prize yet, and I think it will be the honor of all scientists and every medical students and also medical authorities here. What is your advice or principle of achieving this?
Well, first of all, my advice is that should not be a goal. The goal, if you're interested in science, is to experience the thrill of discovery. That is the goal. That is the greatest reward. What happens later on, you have no control over. I know many great scientists who've made fantastic discoveries who don't win a Nobel Prize. They have to keep going because of the excitement of discovery. That's what got me into science. I started university with the intention of going to medical school, but in my first year in university, I had the opportunity to work in a research lab, and that changed my life. And the rest of my life has been devoted to that thrill of discovery. The moment that I saw that image on the electron microscope screen of a cell that was chock full of these vesicles, I knew that I would have excitement for the next 20 years. It turned out even longer than that, of discovery, because I had a new gold mine to exploit. That's what the thrill of discovery is about. It's not about winning prizes.
That's such inspirational. Wow. Thank you so much for that.
I literally got goosebumps from your answer on that. I think as a scientist myself, I think that the hype of seeing the data and then actually excite and keep us forward. I think we have another question at the back as well.
Okay, so thank you so much for your wonderful talk. I'm from Biomedical Science and Biomedical Engineering Department. Actually, I have two questions, but my second question was asked by the previous student already. That's about your motivations or inspiration when you work in your career, right? I think you did not expect that you will get the Nobel Prize. So what is your motivation?
Anybody who expects that is delusional.
Right. Okay, so I still have one more question. So can you give us for the foresight on the genetic engineering and the precision medicine? Because I think nowadays we talk a lot of the precision medicines. So can you give your foresight on this?
Well, I'm not sure what more I can say that I haven't already said. I think for genetic diseases, there will be very effective treatments that either revert mutations or replace genes with normally functioning genes in those tissues that are particularly susceptible to a genetic malady. And sickle cell anemia is just the first, but I guarantee you there'll be more. In fact, it's now possible, going back to the example that I gave of making one HIV resistant. You don't have to go to the embryo to do that. You could take white blood cells, you can take blood from a donor and culture the cells in the laboratory and delete the gene for the receptor for HIV and introduce those cells back into a patient. This could be done for people who actually are infected with the HIV virus, even as they're being treated with antiretrovirals. You could genetically engineer lymphocytes, reintroduce them back into the patient, and then if they took root, you could stop the patient from bothering to take antiretrovirals, and those cells that had lost the viral receptor gene will now populate the bone marrow, and the patient should survive. In fact, there are already inadvertent experiments that have been done. There are 2 patients in the world who've been cured of HIV infection. They both had bone marrow transplants from donors who happened to have a natural mutation in the HIV receptor gene. And those 2 patients no longer take antiretrovirals. They're cured. This could be done by treating lymphocytes cultured from a patient. So, I mean, that's another example, but, you know, it's not limited by that. I'm sure there'll be dozens of examples in the years ahead.
Yeah. Well, this is very exciting indeed to have. So I think genetic engineering, you know, can push very far, and then that is one of the examples, and then you actually provide us many of the examples already. Well, we have one more question. I think this would be the last question, actually.
Good evening. I'm a medical student.
Can you stand up a little bit? Yeah. ขอบคุณครับ
Okay. Good evening, sir. I'm a medical student, year 1. So my question is quite not about scientific, but I want to ask you that to succeed the goal, have you ever lost something in your life?
Have I lost something in my life?
Yeah.
No.
I mean, like, people who have successful, like, they need to
sacrifice something.
Yeah, sacrifice something.
Oh, I see.
As like
I see.
Family.
Have I had to make a sacrifice? That's a great question. So some people look at a scientist like me. I'm very busy all the time, and they say, I don't want to live like that. I want to have a life. I want to have work-life balance. I'm here to tell you, you can have work-life balance and still be a successful scientist. I had 2 children. I had a family life. I was there for dinners in the evening with the children on the weekend. In order to have that kind of balance, you have to be well organized. And that means when you come in during the day, you actually have to have a plan. You have to write it out in advance and make a list. This is what I'm going to do today, and then get it done. Instead of, you know, playing games or talking excessively with people or posting a picture of your pet on your Facebook homepage, you have to be organized. And if you're organized, you can get a hell of a lot done and still have a good family life. So I didn't feel I had to make any sacrifices to do what I wanted to do.
Thank you very much.
Perfect. I think thank you very much, Nakab, Professor Randy Schekman, for your very engaging discussions now. Could you please remain on the stage still? We still have a lot to give you.
Okay.
Now, may I invite Nakab, Assistant Professor Dr. Niwat Krao Padap, Nakab, PhD President, on the stage to present the token of appreciations on behalf of Prince of Songkla University. Kab. Aww. Chocolate koi. Oh my gosh. Edible chocolate Nobel Prize koi. Right, so next may I invite Associate Professor Reungsak Leetanaporn, the Dean of the Faculty of Medicine, to present the token of appreciations on behalf of Faculty of Medicines. Again. And next, may I invite Mr. Uwe Morawetz to take the photo together with the Dean, the President, and the Professor as well. Yeah, can you look at the camera in the front? That's perfect. And next, can I invite the Dean of Faculty of Science, the Dean Faculty of Pharmaceutical Sciences, and the Dean of Faculty of Dentistry to join the stage to take the photo as well. And also the representative of Faculty of Nursing as well, please. Perfect. So if four of you still on the stage, and can I invite the Dean to come down and the PhD Medical Executive Team join the stage to take a photo, please. And this actually would be the last official photos. So is any one of you want to like to take a selfie with the professor? You can from your seat, or you can come at the front and take a selfie as well. Ladies and gentlemen, as we reach the end of today's event, I extend my deepest gratitude to all of you for your presence today to join our event, not just our today event, for the previous talk as well. And I wish you that you get inspired and get motivated from this talk, and has been as Faculty of Medicine and Prince of Songkla University's honor to hold this event. Until we meet again, thank you for joining us and have a wonderful the rest of your day. สวัสดีครับ ขอบคุณมากๆครับผม